To look at the internal anatomy of the sheep brain, what we're going to do is make a cut down the midline. So you're going to place your brain in the dissecting pan, dorsal surface up, as you can see here. Now use a scalpel to cut along the medial longitudinal fissure, extending the cut down to the cerebellum and spinal cord to separate the brain into two longitudinal halves. Okay, so we're going to start here. And we're actually going to kind of use the fact that the brain has a bit of a separation between the two sides to sort of pull it apart a little bit as we cut. So we're carefully trying to cut, making a nice clean cut between the two halves of the brain. So I'm sort of pulling it apart. Together, right? The two sides are only held together sort of very loosely. Okay, so I've made a bunch of cuts. I'm going to go back over here and cut some more down the middle. Now I'm cutting into the cerebellum. And we're almost done, so I'm going to cut one more time here. And I've made a reasonably clean cut between the two sides. So now we have two halves that are nearly identical, as you can see here. Okay, so we're going to take one of the halves and take a look at what we can see with it. So I'll use my probe here to sort of point things out. So again, this is the as far anterior as you can get. And this is posterior. So we're going to first look for the corpus callosum. So it has nerve fibers or axons that are myelinated. And so that's this structure here. You can see this thing coming around here. That's the corpus callosum. So that connects the two halves. You can see that there's an identical structure on the other side. And so that allows for information to be sent back and forth between the two halves of the brain. Now you notice that there's a little bit of an opening right beneath the corpus callosum on one side. So this is what's called a ventricle. It's called a lateral ventricle. So I can put the probe in and sort of open that up a bit. So that's a fluid-filled ventricle. It's filled with cerebral spinal fluid. Okay? So the reason for this on the other side is simply because there's a kind of septum. So there's a thin membrane that separates the two ventricles. Now there is some communication between the two ventricles, meaning that there are small holes, microscopic holes that we can't really see. I'm going to go ahead and cut the septum a bit. And if I do that, we should be able to see the lateral ventricle on the other side. Okay, so it's in here. It's a little harder to get to, but it's in here. Oh, there we go. So here's the lateral ventricle on the other side. So that's a fluid-filled cavity filled with cerebral spinal fluid. So we have them now on both sides, as you can see here. Okay, and above that you have the corpus callosum, which starts around here, goes all the way over here. Okay, what's the next thing we want to look for? So a very good structure to look for is the cerebral cortex. So you can see above this in the cerebrum, there's the cerebral cortex. You can see this cortex means bark, so it's this outer layer here. That's the cerebral cortex. So there's a lot of processing of information going on in the cerebral cortex. Let's dive a little bit deeper into the brain. We can see again, there's another fluid-filled cavity here. Okay, and this round structure is a really important one. It's the thalamus. It's actually a bilateral structure. So what we're seeing here isn't really the whole thalamus. It's just the part of the thalamus that's medial that connects the two roundish thalami on the two sides. Okay, but this, we can consider this the thalamus. It's certainly part of the thalamus. It's connecting the two halves there, and it's almost it's a fairly good, very circular structure there. So that's the thalamus. Now, a very important structure is the thalamus is for relaying information, such as sensory information, processing of sensory information. It also relays information from one part of the cortex to another part of the cortex. But there's another structure that's also very important. That's called the hypothalamus. So hypo means below. So the hypothalamus is located right below the thalamus. Now let's go back to our brain. Let's put it back together for a second. Remember, we had the optic chiasm, which is formed by the X. The optic nerves are missing, but we have these optic tracts here. And right behind that, I told you that that's where the pituitary gland would be attached. And sometimes you may be lucky enough to have it attached. So if we know pituitary gland. Now, if we look at our brain here, doesn't really matter which side we look at. Right there, that's where the pituitary gland would attach, and so that's just below the thalamus. So that's the area called the hypothalamus. So information. So there are neurons within the hypothalamus that will release neurohormones and so forth. In the pituitary gland, that affect the release of hormones from the anterior pituitary gland. There are also neurons within the hypothalamus that will have axons that go into the posterior pituitary, where they release neurohormones. What else can we talk about? So another important structure, if you go, now this is anterior over here, this is posterior over here. So posterior to the thalamus is a very important gland, and that's the pineal gland. Okay, so that's important in regulation of things like circadian rhythm and so forth. So what else do we want to look at? We want to look at, let's next look at the cerebellum. Okay, so that's part of the brainstem. Let's look at the brainstem again, so look at all the various parts. So we'll start at the very tip here, at the very end. This is actually spinal cord, but then the brainstem consists, which is here. And then that ends where we get to about here. This is the pons. There's kind of a bump here that tells us we're at the pons. We then have the midbrain structures up here, which consist of, and it's a little hard to see now because I've cut this apart, but there are these two processes. Now, well, let's look at it this way. This is the cerebral peduncles. I kept calling them cerebellar peduncles, which of course is incorrect. These are the cerebral peduncles. So that's part of the midbrain. So then if we look at this from the side, we know that this is a midbrain structure here. And actually we looked at this earlier, in an earlier video, when we were looking at the external structures. We could see the superior and inferior colliculi, and again that was a midbrain structure. So now we know that all this from here to here roughly is midbrain. Okay? And again, we have the thalamus with the pineal gland. We have the hypothalamus right here. We have the optic chiasm here. We would have olfactory lobes if they hadn't been torn off, so they would be here. The olfactory system is kind of interesting because information can go directly from the olfactory system into the cerebral cortex for processing. It doesn't have to go through the thalamus. Again, here we have a lateral ventricle. We have the corpus callosum, as I mentioned earlier. If there's anything else we need to look at. So we've done most of the. Let's go. I started to talk about the. Sorry, I got distracted. We want to go back here and look at the brainstem. So part of the brainstem, of course, starts with the midbrain. It goes to the pons, which is here. Then all this is the medulla. But part of the brainstem, of course, is the cerebellum. So the cerebellum's involved in things like fine motor movement, motor planning, and things like that. So this is the cerebellum, and what's really incredible about the cerebellum is that it has this really interesting branching structure. You can see this side of it. Now this is because you have white matter. White matter is consists of axons of nerve cells or neurons, which are myelinated. So they have this sort of fatty substance that wraps around the axons. So fat or lipid is white. So that's why you have this white color. And so it looks. So this is called the arbor vitae, because it looks like little trees in there. So it's a very interesting structure. So that's something you definitely want to look for. Let me see. I feel like I'm missing something. Oh yeah, we're gonna now do one last thing, which is kind of interesting. Is to take this. We have two sides, so we're gonna basically take one side, and we're gonna go ahead and make a cut across through here. And what I want you to look for is, and we could use either piece. Here's a good. This piece is fine. What you want to look for is, again, the white matter. So you can see that the cortex on the external surface here is gray, and that's because it has a lot of cells that are, so a lot of information processing is going on locally. So you have neurons and they're connected to each other, but they're not necessarily myelinated. They don't have axons that are myelinated. But then in order to send information from one part of the cortex to another, you want things to travel quickly. And so it turns out in order to send signals quickly, you want myelinated axons. And so they're wrapped again with this fatty substance or lipid substance, which is the myelin. And that's going to have a white color. So hopefully that shows up in this video here. You can see that this is sort of a whitish color in here. In this sort of branching structure, and that's the white matter. And then surrounding that on the outside is the gray matter.